Skin glands, sebaceous, eccrine, and apocrine glands. Introduction. The human skin has several types of exocrine glands, which release their biochemical products onto the skin surface. All skin glands consist by a secretory compartment, the gland or coil tubulus, and an excretory part, the duct ductus. Skin gland cells are of epithelial origin, but their secretory compartments are located at different depths in the dermis. Three major types of skin glands are recognized according to their product, the excretory function, and the location. Regarding their product, skin glands are classified into glands secreting sebum, sebaceous glands, and sweat glands. Concerning their secretory function, skin glands are classified into holocrine glands, whose fully differentiated secretory cells burst and release both the cytoplasmic content and the cell membranes into their ducts, and merocrine glands, which excrete their product via exocytosis from secretory cells. Regarding the location where their ducts release their product, the ducts of sebaceous glands, in most cases, and apocrine sweat glands excrete their products into the hair follicle canal, and the eccrine sweat glands excrete directly onto the skin surface. Sebaceous glands. Sebaceous glands are multilobular structures that consist of acini connected to a common excretory duct and are usually associated with a hair follicle. The sebaceous glands excrete lipids by holocrine secretion. Human sebum contains squalene, cholesterol esters, wax esters, and triglycerides. Sebaceous glands are regulated by several molecules, among them androgens and retinoids. Anatomy of the sebaceous glands. Human sebaceous glands are multilobular structures of epithelial origin that consist of acini connected to a common excretory duct, Fig. 6:1. Sebaceous glands are composed of sebocytes, which are lipid-producing uniquely differentiated epithelial cells. On the other hand, the sebaceous duct is lined by undifferentiated keratinocytes and is usually associated with a hair follicle which is composed of stratified squamous epithelium. The gland secretes in a holocrine manner. Surrounding the glands are connective tissue capsules of collagen fibers for physical support. This is the anatomy of the sebaceous gland. Sebaceous glands are associated with hair follicles all over the body. A sebaceous gland associated with a hair follicle is termed a pilosebaceous unit. The glands may also be found in certain non-hairy sites, including the eyelids, meibomian glands, tarsal glands, the nipples, Montgomery glands, areolar glands, around the genitals, Tyson glands, and the mucosa, lips, gums, and inner cheeks and genitals, Fordyce spots. Fordyce spots open and release their content directly to the epithelial surface. Only the palms and soles, which have no hair follicles, are totally devoid of sebaceous glands. The largest glands and greatest density of glands are located on the nose, followed by the face and scalp. The development of the sebaceous glands is closely related to the differentiation of hair follicles and epidermis. At the 10th to 12th weeks of fetal life, a stratum intermedium becomes apparent, and at about the same time developing hair germs are quite distinct. By 13 to 16 weeks, the glands are clearly distinguishable, arising in a cephalocaudal sequence from bulges, epithelial placodes of the hair follicles. Despite continuous differentiation of its cells, the sebaceous gland can be regenerated by the reservoir of stem cells in the hair follicle bulge. The left picture shows different stages of sebocyte differentiation. Under H and E stain section. The right one show differentiation stages of human sebocytes in tissue, left, and in vitro, right. On the left branch, when SHU, sonic hedgehog, and MYC signaling are increased, and WNT signaling is decreased, the stem cell differentiates into a sebocyte, a lipid-producing cell important for sebum secretion. On the right branch, when WNT signaling is upregulated along with LEF1, the stem cell differentiates into a hair cell, contributing to hair follicle formation. Genes and their proteins and lipids reported to be involved. in sebaceous differentiation and maturation. Physiology of the sebaceous gland. The sebaceous glands exude lipids by disintegration of entire cells, a process known as holocrine secretion. As sebaceous gland cells are displaced into the center of the gland, they begin to produce lipids, which accumulate in droplets. With approaching the sebaceous duct, they disintegrate and release their content. Only neutral lipids reach the skin surface. Proteins, nucleic acids, and the membrane phospholipids are digested and are apparently recycled during the disintegration of the cells. Lipid composition of sebum. Sebum production is a continuous event. Human sebum contains a mixture of nonpolar, neutral lipids, mainly triglycerides, wax esters, squalene, and smaller amounts of cholesterol and cholesterol esters. During passage through the hair canal, bacterial enzymes hydrolyze some of the triglycerides. The wax esters and squalene distinguish sebum from the lipids of human internal organs. Function of sebum. Sebum in humans was initially considered to solely cause acne. Subsequently, it has been suggested that sebum reduces water loss from the skin surface and functions to keep skin soft and smooth. Sebum has later been shown to have mild antibacterial action, protecting the skin from infection by bacteria and fungi because it contains anti-inflammatory lipids and immunoglobulin A, which is secreted from most exocrine glands. Sebum flow to the surface of the skin may provide the transit mechanism necessary for vitamin E to function. Innate immunity. Antimicrobial peptides including cathelicidin, psoriasin, beta-defensin 1, and beta-defensin 2 are expressed within the sebaceous gland. Functional cathelicidin peptides have direct antimicrobial activity against Propionibacterium acnes. In addition, free fatty acids in human sebum are bactericidal against gram-positive organisms. With the expression of innate immune receptors and antibacterial peptides, the sebaceous gland may play an important role in pathogen recognition and protection of the skin surface. Factors regulating sebaceous gland size and sebum production. Sebaceous glands are multifactional, regulated among others by ligands of sebaceous gland cell receptors, such as androgen and estrogen receptors, peroxisome proliferator activated receptors, PPAR, and liver-X receptor, LXR, neuropeptide receptors, retinoid, and vitamin D receptors. Androgens. Sebaceous glands require androgenic stimulation to produce significant quantities of sebum. The weak adrenal androgen, dehydroepiandrosterone sulfate, DHEAS, is probably a significant regulator of sebaceous gland activity. Retinoids. Isotretinoin, 13-cis-retinoic acid, 13-cis-RA, is the most potent pharmacologic inhibitor of sebum secretion.